my name is Marianne Borderes. I'm a bioinformatics software engineer at MatPharma, a company that develops microbiome ecosystem therapies in oncology. I will present this poster entitled Benchmarking of Shotgun Metagenomics Analysis Results Obtained with State-of-the-Art Optimized Pipelines on Simulated Gut Microbiome Samples. In this study, our objectives were to benchmark our gut microbiome analysis toolset performance and to identify the main drivers impacting the results. With this in mind, we have compared the taxonomic and functional results obtained with a gut proprietary pipeline at MatPharma named mgrunner with 3 optimized and routinely used analysis pipelines. We used 2 simulated metagenomic datasets, one to evaluate the taxonomic results and one for the functional results. As shown in Figure 1, to create the dataset to compare taxonomic results, we selected genomes of strains belonging to 41 species, mostly associated with the human gut microbiota. We then created 40 samples by simulating reads from the genomic sequences of the selected strains and generated expected evidences at all taxonomic levels, along with expected values for alpha diversity indexes, such as species richness, and for beta diversity indexes. For the functional analysis, we selected 43 genes belonging to different organisms. We functionally annotated these genes and then simulated reads from gene sequences to create 3 samples. Based on the evidence of genes and their functional annotation, we generated cow and CASAM's expected evidences along with gene richness values. We then shared these 2 simulated datasets with 3 different pipelines owners and collected their results. To evaluate these results and the ones generated by mgrunner, we used several evaluation metrics presented in Table 1 to compare each of the observed values with the pre-computed expected values. We distinguished 2 main types of metrics. The first type corresponds to evaluation metrics computed directly between expected and observed evidences, including deviations of relative evidences, recall, precision, FN score, Brickertis and Jacquard similarities. These 2 latter beta diversity similarity metrics are complementary since Brickertis uses the evidence of taxa and Jacquard uses only their presence-absence. The other type regroups deviations and absolute deviations between expected and observed alpha and beta diversity indexes. All these metrics were used to benchmark taxonomic results, but only deviations of relative evidences, recall, precision, and FN score were used for functional results. We computed these metrics for each sample and then represented the mean value across samples. Figure 2 shows the result of our comparison for the taxonomic analysis at the genus level in the top panel and the species level in the bottom panel. Each pipeline is represented by a different color, and the optimal theoretical values are represented in black. Metrics with an optimal value of 0% are represented in the left panels A and C, where we have all the deviations, and metrics with an optimal value of 100% are represented in the right panels B and D, where we have, for instance, the mean Brickertis similarity and the recall. Before we dive into the result, it is important to note that because we apply different rarefaction thresholds, each external pipeline's result can only be compared to mgrunner's and 2 optimal theoretical results. Let's first focus on the genus level. As we can see in Figure 2a, pipeline B and C, represented in green and yellow, showed greater deviations of relative evidences and of evidence-based alpha diversity indexes as compared to mgrunner, represented in blue. These results can be explained by main differences from mgrunner and the used bioinformatics analysis strategy and database. Inversely, pipeline A, represented in red, which used a different strategy but a similar database, had lower deviation values than mgrunner for these latter alpha diversity indexes. It had, however, a larger genus richness deviation value, not shown in this poster, which was mainly caused by a more permissive taxa abundance filter threshold applied as a post-processing step. If we now look at Figure 2b, we can see that the best trade-off between all the metrics represented in this figure was reached by mgrunner. However, pipeline A in red was better at recovering the complete expected community structure, as we can see with its Brickertis similarity and recall values, but showed lower performances in precision and Jacquard similarity. In contrast, pipeline C in yellow showed better precision and Jacquard similarity and also genus richness deviation values, but lower recall and Brickertis similarity values. At the species level, we globally observed a shift in the distribution of values, indicating that all pipelines had a lower performance on several metrics in comparison with the genus level. These can, for instance, be seen on the event score or the deviation of relative evidences. Although the trends were overall similar, we observed that the performance gap between genus and species levels was not identical between pipelines. To conclude with the taxonomic analysis, we overall observed an imbalance between precision and recall, which was easily noticeable with pipeline A and B, and between evidence-based and presence-absence metrics, which was well in evidence by the difference of performances measured between Bracketis and Jacquard similarities. Overall, mgrunner gave very satisfactory results compared to the three other pipelines and maintained the best balance between precision and recall at both taxonomic levels. Concerning the functional analysis, the benchmarking of results was challenging. We were only able to compare mgrunner to pipeline C and only partially to pipeline B due to missing outputs for these external pipelines for expected functional categories. We observed that mgrunner obtained the best performances on all computed evaluation metrics. Pipeline C, which has a different database version, had the closest results to mgrunner, and Pipeline B, which has a different analysis strategy and database, had the least precise. These performances can be explained by the fact that we computed the expected results for the functional categories using our functional database. Thus, to address this limitation, we believe more effort should be made to create simulated datasets with comprehensive and database-independent expected functional annotations. To conclude, mgrunner held a good global position as compared to the evaluated pipelines. It showed the best event score values, demonstrating the best trade-off between precision and recall. We have also identified paths of improvement. As expected, the use bioinformatics analysis strategy and database had a strong impact and performances, but so did the taxa abundance filter threshold. Ultimately, this study shows the importance of evaluating complete analysis pipelines with optimized parameters using reference, taxonomic, and functional standard datasets to properly assess the quality of obtained gut microbiome profiles and the limit of analysis pipelines.